Hello, my name is Hannah, and my project was on the sex-dependent dietary impacts on transposable elements epigenetic silencing. Transposable elements, or TEs, are widespread genomic parasites which replicate themselves and spread across the genome. To combat against their harmful spreading, many eukaryotes deposit repressive epigenetic marks such as H3K9 dimethyl or trimethyl to silence the TEs. However, sometimes these repressive epigenetic marks can spread to nearby genes, perturbing their function and shaping the evolutionary changes of the organism as a whole. Connecting both principles, I'm interested in observing if altered diets impact the epigenetic silencing of TEs. To directly observe, we have a lab-developed transgenic strains, which is going to have the mTERI marker inserted on chromosome 2, causing the flies to glow red under the fluorescence microscope, but we specifically focused on the eyes. And the two lines we used is no TE, which is our control, and 1360, which is going to have a 1360 DNA transposon inserted 269 base pairs away from the mTERI genome. And the varying degree of TE silencing is reflected as different mTERI fluorescence intensities. We took those two lines, kept them on the ketogenic curcumin and the corn-fed food on the dietary condition for 3 generations, took 30 males, 30 females, and imaged their eyes. We chose ketogenic and curcumin dietary conditions because they are well-known histone deacetylase 1 inhibitors. And based on the results here, curcumin and ketogenic diets across all 3 replicates showed sex-dependent effects, where the females had an increase in normalized intensities, indicating a decrease in TE's epigenetic silencing, and the males showed a decrease in the normalized intensities, indicating an increase in TE's epigenetic silencing. This raised the question of what mediates the sex-dependent effects in TE's epigenetic silencing. And to directly observe, what we did was we utilized the XX attached Y and XO crossing system, where we used the BDSC 9464 cross, where we have the X attached Y, and that means the X and Y chromosomes are physically fused together, so they would segregate together during cell division. And what we did is we kept them on the food for 3 generations, and we ended up getting the XX attached Y females, where all the Y chromosomes is present in females, and none of the males had the Y chromosomes, so they would be XO or sterile males. We imaged their eyes, and what I concluded was that the XX attached Y females and XO males respond to the curcumin and ketogenic diets similarly, meaning there is no sex-dependent effects. That helped us conclude that the sex-dependent dietary effects in T's epigenetic silencing that we observed beforehand is possibly due to the presence of the Y chromosome in male background. Further replication and different crosses are going to be needed to perform to make a solid conclusion. Thank you.